first of all. All right, now I'm recording, and then I want to go over what you're going to do in lab tomorrow so that they don't have to do it. And so you can just immediately kind of get into it and get going on it. So I'm going to share my screen. Here is the light reaction. All right, let me. Okay. Okay, so the light reactions of photosynthesis. So it's really kind of cool. What you're going to be doing tomorrow is actually manipulating the light reactions of photosynthesis by providing different environmental parameters for the light reactions to take place. And so we're going to see that you're going to be able to either have the light reactions cook along at a really rapid rate, or by certain manipulations, you're going to cause it to slow down. And then we'll be able to compare what's happening under different environmental conditions to the rate of photosynthesis. So let's just, you're going to be, oh, by the way, you're going to be working mostly in the dark. So this is a picture of the lab from a few semesters back. There's Claire, I remember Claire. So we have desk lamps that will be set at each table, but the overhead lights will be off. Now, but you know, the desk lamps are going to do the job, so don't worry about it. So what we're going to do is we're actually going to think about what's happening in the chloroplast. So we've already looked at chloroplast structure. We know that the internal membrane system, the thylakoid membrane, is where we see all the chlorophyll embedded into that membrane, and that's where the light reactions of photosynthesis are going to take place. So that's the next topic in lecture, by the way, is looking at light and chlorophyll and the light reactions. So you're going to see these again, but the big picture idea here is that with the light reactions of photosynthesis, we're going to be looking at the idea of an electron transport chain process. And they don't really show it here. That what you're seeing here is the big picture idea of, in total, what's going on with photosynthesis. Part one is the light reactions, where they're going to be making NADPH and ATP, and that's necessary because part two of photosynthesis is the biochemical reactions, where you see the CO2 coming in and getting fed into the Calvin cycle to make sugars. So they go hand in hand, and when you look at the light reactions, you're going to see this many, many times when we get into it, and we're going to know all about this. But basically, this is the electron transport chain process that you're seeing. And let's start over here on the left. We have PS2, and then over here, PS1. Those are photosystems. We'll get into those. But basically, there's chlorophyll in these photosystems that will be absorbing light energy, which is light breaking those photosystems. And when photosystem 2 gets hit with light, it actually creates a situation where they can strip electrons off of water, which is very, very challenging. And so it needs lots of light energy to get that to happen. And so electrons will get stripped off of water, and then the black lines here are showing the progression of the electrons as they move down the electron transport chain process. And so as you see, there's different players in the game here, but basically the electrons will eventually reach photosystem 1, which is also getting hit with light. And then the electrons continue on, and we say that the final electron acceptor is NADP+. All right, so the ultimate electron donor is water over by photosystem 2. The ultimate electron acceptor is NADP+, and then it gets reduced to NADPH. And then as this electron flow is happening, you create a situation where it allows for protons to get pumped across the membrane during this process. And so down here you see protons building up in the lumen portion of the thylakoid membrane. Eventually they'll diffuse back, and as they do that, they go through the ATP synthase, and it's a little molecular motor. The flow of protons creates a torque force that allows those subunits to rotate in such a way that allows for ADP and inorganic phosphate to react, and then you get your lovely ATP. All right, so when we do this experiment in the class, it's called the Hill reactions, named after Dr. Hill, because I'll come back to that slide in a minute. He is looking, he devised a way of how do we track the light reactions, and then let's manipulate it and by giving it different environmental parameters to see what happens to the rate of photosynthesis. So he's credited with that. So basically, we're looking at this idea of how do we measure this process of the electron transport chain cooking along during the light reactions. And the easy way to do that is in the lab environment is if you can come up with some kind of scheme that shows a visual color change. And we do this a lot in microbiology classes too. We look for a visual color change to tell us what's happening. What are those microbes doing? Well, you can do this with plants too, with the chloroplasts. If you can figure out a way to set up an experiment where you can track a visual change, that's really easy for us. We have spectrophotometers to do to be able to measure this. And so he's credited with the idea of coming up with this experiment. Well, what you're going to be doing, and actually they're going to prep this for you. Usually we have you do this, but to save time, our lab preppers are going to do this for you, and you'll be provided with a chloroplast suspension. So normally what we do is we have you grab some spinach leaves, put them in a mortar and pestle, grind in a buffer solution, and then we take you up to the centrifuge and we centrifuge it all, multi-step, and then we get our chloroplast. So all that's going to get done for you. You don't have to worry about that. And so you'll be handed a cold tube of chloroplasts that we extracted from spinach. Okay, well, the problem is NADP plus and NADPH, they're colorless to our eyes. So that's a problem, because what we're trying to show, when you know that the light reactions are cooking along, you should get this transition of NADP plus getting reduced to NADPH. But because it's colorless, there's no easy way to track it. So they came up with a solution which is basically replacing NADP plus with a different electron acceptor that can change colors to our eye and to the spectrophotometer. It can pick it up when it starts accepting electrons. So basically what we're going to do is right before you get going in lab, your instructor will mix up this solution called DCIP. So this chemical, DCIP, is going to be the artificial electron acceptor. We're going to flood the system. We're going to put so much DCIP into that tube that it will be the one taking up electrons as the light reactions are cooking along. And so DCIP is a good chemical to pick, because you'll see if you are not too busy and you'll look up when he's mixing the chemical into some water, it's this deep, beautiful blue, really deep blue. All right, so it's blue when you first mix it up. You're going to put a certain aliquot of that into your reaction tube along with your chloroplast suspension, but you put so much in, it kind of like floods the system, that there's so much DCIP that as you start the experiment, it's the DCIP that will get reduced, and you'll see enough of a reduction over time that it's going to start out blue, and then it's going to start going colorless as it accepts more and more electrons. And so that we can track using a spectrophotometer. So as your experiment continues over time, you'll see the spec readings will be high to start with, because blue is a deep blue pigment. And so we're going to take absorbance values at 620 nanometers, and you'll get a high absorbance value. And then as that blue color starts to go away, you'll see the absorbance values drop. And so essentially what we're doing is the second bullet point here. The first bullet point is kind of the big picture idea of the light reactions, where NADP plus is sitting there to take electrons and become reduced to NADPH. But instead we're flooding the system with DCIP. All right, so DCIP essentially takes the place of NADP plus, and the DCIP will continue to get reduced if the light reactions are cooking along. It'll get reduced to DCIP H2 essentially. Okay, so that's the big picture idea of what you're doing, what's happening in your reaction tube. So then to be able to track the light reactions over time, you have to make that happen. So what you're going to do is you're going to have a reaction tube with a recipe of, put so much buffer and the chloroplast suspension and the DCIP. And then what you're going to do is use that spectrophotometer. So each table will have a spectrophotometer that you'll be using to periodically, every 30 seconds, we're going to ask you to take a spec reading. So what you're going to do is at your table, you'll have your desk lamp on, and you're going to have a bucket of ice with your chloroplast suspension. You're going to mix your reaction tube, and when you're ready to actually do the experiment, you'll have the DCIP spiked in, and then you'll take a spec reading right away, and then you'll put the tube underneath the desk lamp so it's getting exposed to light. You'll hold it there, and you need to be timing it. So have your phone timer up or whatever. We'll probably have stopwatches. Everyone seems to have a phone, but you'll need a timer. And you're going to hold that tube under the desk lamp, make sure it's getting bright light from that. And then every 30 seconds, pop it into the spec, take a reading, back out into the light, another 30 seconds, take a spec reading, back into the light. So you have to do this quickly. You can't hem and haw. Once it's into the spec, quickly take the reading, get it back into the light as soon as you can. So one reaction tube is going to get multiple spec readings off of it because you have to track over time what's happening in that tube, okay? So it's exposed to light, 30 seconds, take a spec reading, back into the light, another 30 seconds, and you hopefully are going to see this drop in the spec readings over time. And so we usually do it for a couple of minutes, and hopefully you'll see this nice progression of a decrease in the spec reading, maybe all the way to 0, which is fine, that can happen, but not always. So this is kind of the scenario where you'll have your spec. I think we have the new ones, these are the older ones, but we'll have some, hopefully the new spectrophotometers and the desk lamp. And there's Claire taking the tube, and she's popping it either back under the light or about to take a spec reading. And so if you do that, and if your reaction tube is working correctly, you should get some data that might, and the numbers can vary, but the trend should be similar to this, where you see at time 0, you start at a high absorbance value, but you can see over time it starts to drop lower and lower and lower, all right? Then you can have your laptops out, and someone could have Excel up and running, and you can start plugging these values in and charting it using a line graph to see how things are comparing. So what we're going to do is, once you figure out that your reaction suspension is working correctly, everything's good to go, then you'll get to do some manipulations. And so we're kind of paring back, we're eliminating one of the things we normally do, but we've kept everything else. So one of the ideas would be, how much light is necessary for rapid photosynthesis? So some tables will be challenged to do this experiment at different light intensities. So you won't be using the actual desk lamp. We're going to give you an old-fashioned slide projector with a beam of light that you can stand in front of at different distances in order to create different intensities of light. So if you hold it right up to the slide projector, it's going to be really bright light. We're going to give you a light meter, and you'll be able to calculate how much foot candles or lux you're getting at that tube, how much intensity is hitting the tube. And then you'll stand at different distances from that light to create dimmer conditions, for example, as you move away from the light. And so you'll stand at a certain point. We usually put tape down on the floor so you know where to stand. You've taken a light meter reading, and then you will hold your reaction mixture right there, 30 seconds, pop it into the spec, take a reading back to that spot, another 30 seconds. And you'll do it at different distances from the slide projector beam of light. So some of you are going to get to do that. So you're actually asking what happens to the rate of photosynthesis when you have different light intensity hitting on the chloroplast suspension. Another thing that we do is we have another slide projector with that white beam of light, but then we put in front of it different light filters with different colors of light. So now you're asking, what quality of light? Do they like red light, orange light, yellow light? What quality of light, color or quality, is going to create a very fast light reaction process, or maybe a terribly slow one? And so you'll be making up these reaction tubes again, and you'll stay at a set, you'll have to make sure that your foot candle stays consistent between whatever quality of light you're working with. So you'll need a light meter for that too, because you want to keep that consistent. And it's so that variable is controlled for. The only variable is what color of light is the suspension getting hit with. So some of you are going to get to do that. And please be very careful with these. They're like blocks of glass that are different colors. It's actually an antique set that I'm having you work with. Comes in this wood box. I inherited it from a former physics professor, Dr. Fowler, who's retired now, but I always used to borrow it from him when I first started teaching here, and he rarely used it. So he's like, why don't you just keep them? But when I looked at the leaflet inside, they were made shortly after the World War. What is it referring to? If it says something, the World War. World War I. So they were made between World War I and World War II. So they're antique. So please be careful. They're fun to work with, and they work really well for this experiment. So some will get to do that. And then the other thing, another table will get to do an experiment where you add in to your reaction tube an herbicide. So this herbicide is called DCMU. It's a common herbicide. And so an herbicide is to spray out into your fields if you have weeds. So a weed is, by definition, a weed is just a plant growing somewhere where you don't want it to grow. And it can be anything really. But if you're trying to grow corn and a bunch of ragweed shows up into that field, you want to get rid of the ragweed. And so you'll hit it with an herbicide. So we're going to do this. What happens to the chloroplast and the rate of photosynthesis when you expose them to different concentrations of an herbicide? And we'll look at how that impacts the rate of photosynthesis. All right. So let's say the other one we cut out because it's a little too fussy and it takes too long is this one where we look at different temperatures on the rate of photosynthesis. So I've given you some mock data here just to show you how you're going to end up graphing this out and thinking about your data. So in this experiment, we had the chloroplast suspension sitting into a cold ice bath of 4 degrees Celsius. And then another setup was at 10 degrees, 15 degrees, 30, 40, and 50 degrees Celsius. So these reaction tubes were held at these temperatures under the light, and then spec readings were taken every 30 seconds. So whoever, so there's 1, 2, 3, 4, 5, 6 different reaction tubes that this group made up. And each tube got, this particular tube was kept at 4 degrees, but every 30 seconds were the absorbance values were taken. So when you graph out your data, you're going to have something like this, where you have many lines on one graph, so you can compare what's happening with your different manipulations. And so you can see here, as you start out at time 0, you probably will see some different absorbance values. They're not all going to be the same, but that's okay. What you're looking for is the slope of the line, essentially, as you work your way from left to right. If it's a steep slope, like the yellow line here, which is 30 degrees Celsius, that means the rate of photosynthesis is cooking along quite nicely. You have DCIP getting reduced quite nicely. After 120 seconds, it's almost 0, and 150, it is 0. It's gone down, the color went all the way colorless. On the other hand, you see this orange line at 10 degrees Celsius, and then the 4 degrees Celsius, you see how shallow that slope is? It's like barely doing anything. DCIP is staying pretty blue, which means you don't have a lot of DCP getting reduced to go colorless. So you're slowing down the rate of photosynthesis.
So obviously there are plants that photosynthesize at temperatures below that. So are the adaptations that they have to photosynthesize at the temperature in the chloroplast expressed there?
Yes, many of them are part of the light reaction apparatus, the photosystems, and and the membrane too. They have some special adaptations for their membrane to function better under cold. Yep, absolutely. But spinach, not so much. Right. Spinach can handle some cold, but in general, they're not super well adapted. So at 4 and 10, you see this pretty shallow line because they're just too cold, and they're not able to do the light reactions very well. Same with the green line here, you see is pretty shallow. That's at 50 degrees. So, your body's at 37 which is 98.6 degrees Fahrenheit. 50 degrees is hot, it's hot, and probably a lot of proteins have been denatured and fallen apart, and there's a bunch of proteins involved in the light reaction, so this is a pretty horrible situation. So it's not too surprising that, again, the light reactions, it didn't really do much of anything. So when you graph out whatever experimental manipulation you're doing, whether it's light quality, light intensity, or the DCMU, you'll end up with graphs like these, and you're doing the comparison amongst the different, like, you know, the different colors of light, the different intensities of light, or the different concentrations of the herbicide. So that's what you're going to be doing. Questions? Okay, so at each table, you'll have to be coordinating amongst yourselves of who's doing what. And, you know, you will be making up several reaction tubes, because each reaction tube, you know, so say you have to hold it at 20 foot candles light intensity. Well, you're going to have to prepare one reaction tube for that light intensity, and then you're going to have to prepare a brand new tube for the light intensity at, you know, 100 foot candles or whatever. All right, so you're going to be having multiple reaction tubes that you'll have to get up and running. And then every 30 seconds, you'll be taking a spec reading as quickly as you can, and then getting back into putting it under the light of some sort, whether it be the slide projector or your desk lamp. Okay, so when you get in to the lab, when you start this experiment, all the lights go off, the overhead lights go off, and you'll be working with just mood lighting and desk lamp. All right. So this slide set is posted under the lab in EDUCAT, under the lab PowerPoint section. So you have that for reference. And then the handout too, of course, that you'll actually follow, the actual experiment, is posted in the lab EDUCAT page as well. All right. Okay, if there's no questions. I don't think there's any questions on the chat. I didn't see anything pop. So, all right, it's going to be fun. This is a great, fun experiment. You get to be totally manipulating photosynthesis by what you're doing. It's pretty cool to see how that impacts the rate of photosynthesis. All right, I need to get into, we got to go back to looking at the so the people zooming in, do you just see one big blue slide with the word invasion on it? Invasion of root hair infection thread? Is that what you're seeing?
Yes.
Okay, thank you. All right, so invasion of the root hair. And again, I take issue with invasion. That sounds so negative. It's not a negative thing. That I don't know. I don't know what else you would say. The pairing up, but it is kind of an invasion of the root hair in a way. But it's a very special way. It's not an aggressive process here. So the infection thread, what the heck is that? That is just basically what happens is the bacteria need to get into the root tissue. And the easiest way and the most common way for them to do that is through the root hairs. And as we know, most of the time, root hairs are long and slender. And so if you can imagine the bacteria, they get in near the tip of the root hair, and then they move down the length of the root hair. And when you look at it under the microscope, it looks like a thread of bacteria moving down the root hair. And so that's where they come up with this infection thread idea. But the invasion process is very controlled. It's not this free-for-all where the bacteria just bust through and get into the cytoplasm. So let's talk about that. So the invasion step is when the bacteria have to make it past the cell wall of the root hair. So they got to get through that. And so that's a very controlled process. Again, the cell wall is loosened up enough for them to get through, but it doesn't, the bacteria don't obliterate it. It's let me squeeze through the cell wall area.
Dr. Mackey?
Yes?
You said that they don't actually go inside the cell or the cytoplasm?
No, they do not.
Okay, just checking.
Yep, so let's see how that happens. Let's imagine this now. So imagine you're a bacteria that you squeezed past the cell wall, but of course, right past the cell wall, then you bump into the cell membrane. Now, if they were to penetrate the cell membrane, that would alert the plant, you've breached my boundary. I did not permit that, you're a pathogen, I'm going to attack you. So that's not what happens. What happens instead is you have to imagine the bacteria got through the cell wall and then they kind of butt into the cell membrane. And then you have to imagine the lead microbe, the lead bacteria in the little trail here, they push onto the cell membrane and invaginate the cell membrane ahead of them. So if you can imagine the bacteria just kind of pushing that cell membrane ahead of it, and that as that's happening down the root hair, essentially the cell membrane invaginates ahead of the bacteria, as the bacteria is kind of like, you know, the ants that follow in a line. If you've ever watched ants when they're out of their little living hole or nest, yeah, colony, it's kind of that idea, you know, the bacteria end to end to end, and the lead one is just kind of pushing against the cell membrane, if you want to think of it that way. And that cell membrane invaginates down the root hair. And so that, when you look at it under the microscope, we say it looks like, they call it the infection thread. And so if you can think of that and visualize that, truly what's happening then is the cell membrane is never breached, and the bacteria are always surrounded by plant cell membrane material. And so again, it's a very controlled infection. The plant is not allowing for the breach of the cell membrane. Let's take a look at that. So here up top, you're seeing the artist's rendition of, remember early on in the chemical communication process, once the bacteria get to the root surface and they exude out those nod factors, it often causes the root hair to curl and kind of hug up the bacteria into this little area where they can then breach the cell wall, but not the cell membrane. And then they're trying to show how, you know, you have these bacteria in a line pushing the cell membrane ahead of them as they migrate down the length of the root hair. And then of course they will get into the bulk of the root beyond the root hair. And so these are just the parenchyma cells of the root, and they're going into these different parenchyma cells and setting up camp, but again, surrounded by membrane. They are not free floating, swimming around in the cytoplasm, okay? And then here, very nice stained images of the root hairs getting infected and invaded, so to speak. And you can see a congregation of bacteria starting to gather in the bulk root area. Here you can see a very nice infection thread on the right side here, in the dark line here going down the root hair. That's the infection thread. Whoops, that's just cell membrane and the bacteria as they work down the root hair. Some other really nice images, the infection thread with the bacteria. Great image here, where you see the bacteria end to end to end, moving down as a, you know, a line of bacteria down the root hair. And that cell membrane is surrounding them as they progress down the root hair. Another really great image of the root hair and the infection thread. So a very controlled process, as we saw the other day. Lots of chemical communication leading up to this. You have to have both of them sending out chemicals and being able to chemically communicate that this is something they need to or want to engage in, so to speak. And set up this symbiosis. Okay, so the bacteria have moved down the root hair and they go into the parenchyma cells of the root, and they, excuse me, they start to set up camp, so to speak, in these different parenchyma cells surrounded by the cell membrane. And so what's going on there? So they actually live in vesicles, so they're in the cell membrane, kind of buds off to fully enclose the bacteria. So I imagine, you know, the bacteria kind of floating around in a balloon, a vesicle, and that's what's floating in the cytoplasm, all right? So it's kind of like they're on an inner tube, but they're fully enclosed, all right? And they have this vesicle totally surrounding them, and that's what's in the cytoplasm. Some people,
So
go ahead.
is it possible for plants to then like pass on those bacteria through the cytoplasm, through like a seed or something?
typically, I don't think that has happened. You can get other endophytes that end up in seed. They know that, but I don't think the nitrogen fixers do that.
I guess it makes sense they're in the root, but
Yeah. Although, but, having said that, there are endophytes that like live in leaf tissue, but they migrate to the seed when the seed is starting to form, and in the root too. So it's not out of the realm of possibility. We know that endophytes can migrate to the seed as the seed is being formed. Not all of them do that for sure, but there's some very special ones that can do that. That's why seeds, you can surface sterilize seeds, but you often have microbes already in the seed. And they're, the good microbes, the ones that are typically there to help the bacteria or help the plant. So depending on who you talk to, some people might call the vesicles symbiosomes because they're, housing the bacteria, and that bacteria are symbiotic. So just be aware there's some jargon there that you might see. And then other papers I've read, they call it the peribacteroid membrane. So just be aware that there is some differences of what they're, the jargon, but basically it's the same basic idea. There's some kind of membrane material, it's the cell membrane material that buds off to totally enclose those bacteria so that they can be in the cell, but they're always surrounded by that membrane. And then after that happens, you'll see the bacteria, they actually start to grow quite big actually. They're impressive. Most bacteria can change their size somewhat, but these seem to really have a special knack for it. So they enlarge as they're in those vesicles. And when you see that under the microscope, then they'll use the word bacteroids. So don't get too caught up in the jargon when they say that, or if you read that, they're just talking about the bacteria that have now really set up camp and are enlarging to do their job, which is to fix nitrogen.
Does that just have to do with increased availability of nutrients?
Yeah, I mean, it is a pretty sweet setup for these bacteria. They're in the root tissue, and we're going to see, the next bullet point, the vascular connections form. And so now the bacteria are getting fed by the plant. So the bacteria are getting all these lovely sugars from the plant. And so they're getting all this free food. So well, it's not free, but it's a very nice situation for the bacteria. And so you can actually see an increase in vascular connections into these nodules where the bacteria are, so that the plant can feed the bacteria, but then also you need a way to get the fixed nitrogen into the rest of the plant. So it makes sense that these vascular connections are ramped up, and then it's a two-way street. The bacteria are getting fed, but also the bacteria will give a lot of their fixed nitrogen to the plant. So importive photosynthate is the fancy way to say that the bacteria are getting fed by the plant, the sugars primarily, and then the bacteria in return will give a good chunk of the fixed nitrogen to the plant. They'll keep a little for themselves, but the bulk will go to the plant, and they think it's primarily through xylem movement that the fixed nitrogen gets to different parts of the plant body.
What do you mean by vascular connections?
The xylem and phloem is the vascular tissue.
Go from that to the little bacteroids?
To the nodule. So if you were to cut off a knot, well, if you cut it off, you cut the vascular connections. But if you look at a nodule intact in the root system, you'll see the xylem and phloem, the vascular tissue connection into the nodes is kind of ramped up and beefed up. So that nodules, when you look at the nodules, if you dig up roots that have these bacteria in a symbiosis with the plant, these nodules are the home, in essence, of the bacteria. So that if you were to cut this open and look at it under the microscope, the right image is what they did there. So all these greenish things, that's all the bacteroids in the nodule. All right, so it's plant tissue mostly. All right, so what's happening is the plant kind of forms a callus, if you want to think of it that way, to form the nodule. It's just the plant cells going through cell division to form this mass, kind of like a tumor, if that's an analogy that you're comfortable with, but it's not a negative thing, okay? And then if you cut into that nodule, you'll see it's just a home for all these bacteria to do the fixing of the nitrogen. All right. So here, these are the bacteroids you can see again, but I like also that this image captured the membrane material, the vesicle material, or the symbiosome, or the peribacteroid membrane, always surrounded by plant-derived membrane. Okay, so they invaded, they got into the parenchyma cells, they've set up camp, the vascular connections are good to go. Then they're going to start fixing nitrogen. So the bacteria are the ones that are going to take, dinitrogen, N2, and using that very special nitrogenase enzyme, cleave the triple bond, and then you get ammonia. Well, that's not free. It's costly. So,
Dr. Mackey,
yes,
does the nitrogen get into those nodes through diffusion?
I believe so. I don't think there's any other method that they've seen. It's just diffusion processes. I believe so, yes. So this overall reaction, the main thing for you to appreciate here, of course, they have the dinitrogen as one of the substrates, but look at that, 16 ATP for one dinitrogen to get fixed to ammonia. That's a big cost, 16 ATP to get that reaction to happen. So this is not free. However, obviously the benefits must outweigh the costs, because there's plenty of plants that have formed this relationship with these bacteria, and the bacteria are still doing it. So it has to be a win-win. Otherwise they would stop it if it got to be too costly. They've done some looking at, like what is the overall cost? And often when you are looking at the cost to the plant, you estimate how much carbon has to get sunk into this partnership, so to speak. And so that's how they usually portray the cost. And so for soybeans, the cost is 12 grams of carbon is required to fix 1 gram of dinitrogen. And it's just a way to drive home the point. It's costly. The plant has to invest in terms of its resources to get this to happen.
When we say 12 grams of carbon, are we talking about like the entire compound of like whatever sugary?
Yeah, I think they're mainly focusing on, once the carbon dioxide gets fixed into sugar, there's 12 grams of carbon in the sugar, of the sugar.
Are they counting the carbons or like the whole molecule?
They do the carbons.
Okay.
Yeah, it's weird. I know. It's just the way
that
they've
makes
always
sense.
done it. So yeah, so they focus in on the carbon. So basically what that's saying is it's a cost. The plant has to invest to maintain this relationship, but it must be worth it, because this is something that has been going on for millions of years and it's been maintained. So let's look at it down at the level of the actual enzyme that is so unique and allowing for this fixing of nitrogen. So this is the nitrogenase enzyme complex. It's multi-subunit complex that works together to coordinate this activity. So over on the far right, you have the actual reaction of dinitrogen getting converted to ammonia, all right? But what else do we have going on here? Here's your 16 ATP that they say you have to get this going in order to allow for this reaction to happen. So let's go over here to the left. There's this funky molecule called ferredoxin, which actually, it's a protein. We'll talk about it because it's actually part of the light reactions of photosynthesis. So keep that in mind. So ferredoxin is a protein that's very happy to donate electrons to some other reaction that needs it. Sometimes it's the light reactions, sometimes it's this. All right, so ferredoxin is in its reduced state on the lower left here. And what they're showing is ferredoxin is donating electrons to this iron protein. So here you see it's the iron protein is in its oxidized state down low here. It meets up with ferredoxin in its reduced state, and ferredoxin says, hey, I'll give you some electrons. So the electrons get donated to the iron portion of this protein, and that reduces the iron protein, the iron portion of the protein. So what you've just seen here is an electron transport mechanism that's kicking off here. Ferredoxin donates the electron to the oxidized iron portion. That reduces the iron portion. Now that will move over and reduce the molybdenum iron protein. I have a really hard time saying that word. So you see here, the molybdenum iron is in the oxidized state, and it's like, hey, I need some electrons, and the iron protein says, I got some, here you go. And so the electron gets transferred to the molybdenum iron. That gets reduced. And then finally, you see nitrogen, dinitrogen, accepting an electron in this process as the triple bond gets cleaved and gets reduced from dinitrogen to ammonia. So it's a reduction reaction if you look at it. So this is basically a small electron transport chain process in the sense of electrons are getting donated from donor to acceptor. It's not very long, but it works. And the big payoff, of course, is you get dinitrogen fixed to ammonia. But it's really a reduction process when you look at it. It's getting reduced to ammonia. All right, so very unique enzyme. Only prokaryotic organisms have it, and only a subset have it. We're focusing on the bacteria that can do it when they set up camp with the plants in the roots. But remember, some can do it all by themselves. Some bacteria are fine to just be by themselves, and they can do this for their own purposes. And then some bacteria can do it when they want to, so to speak, but can shut it down when they don't need it. All right, so they don't always have to do this. So it's just a matter of, I'm gonna keep talking about the bacteria that are doing this with the plants, of course, because this is a plant class. But one thing about this enzyme, so it's really quite a unique, special enzyme, and it has this amazing capability to push this reaction of taking dinitrogen gas and making ammonia from that. However, there's a big downfall to this enzyme. It's super sensitive to oxygen. So the dinitrogenase or nitrogenase enzyme, it's got some issues with oxygen. So if this enzyme is bathed in too much oxygen, if too much oxygen is around it, it totally breaks it down, inactivates it, denatures it, it's toast. Well, obviously they figured something out to deal with this because we still see plenty of nitrogenase function going on. So somehow they've dealt with this. Well, they have. They have, in this partnership, when you have the rhizobia bacteria, they have, well, in concert with the plant, it's a partnership effort here. They create this oxygen binding protein, a heme protein. So when it's a multi-subunit protein, and it's called leg hemoglobin. So right away you see the word hemoglobin in there, and if you've taken any physiology, animal or human phys, you're like, wait a minute, that's in our red blood cells, and you're absolutely right. Leg hemoglobin, and I'll show you a picture, it's so similar to hemoglobin. It's eerie, it's quite cool. And it actually makes the nodules pink. Like our blood is red, it makes the nodules pink and slightly red. And so it's really cool how similar this is to hemoglobin. Well, it's a partnership process. So it's a multi-subunit protein. And the plant says, I'll volunteer to make the globin part. You know, if bacteria, you go ahead and make the heme part, and then we'll put it all together and get it up and running, and it will do its job of binding oxygen. All right, so leg hemoglobin, this is not the actual nitrogenase enzyme. That's a separate entity. This is a totally different protein, this leg hemoglobin, multi-subunit protein. And so its job is to, just like our hemoglobin binds oxygen, right? It does that too. So leg hemoglobin will bind oxygen, and if it's bound, then it's essentially unavailable to mess with the plant, or mess with the nitrogenase enzyme, I should say. All right, so if it's bound to the leg hemoglobin, then it's not going to negatively affect nitrogenase. So what happens is the leg hemoglobin will bind the oxygen, and just like hemoglobin brings oxygen to our cells and tissues, it's doing that too. It's making sure that the bacteria and the nodules still have oxygen, that the plant cell still has oxygen, but just in a really controlled way. So it will release the oxygen just a little bit at a time so that the bacteria are getting its oxygen needs met, but nitrogenase isn't getting exposed to any extra oxygen. So this is a nice way to kind of sequester the oxygen away from nitrogenase, but still make sure that the plant cells and the bacteria have enough oxygen to do their respiration and breathe, so to speak.
Do you know if non-vascular plants have any kind of like symbiosis like this?
Yes,
They do.
there are some bacteria that have, in fact, I just read a paper. Let me see if I can find that again. Where they don't form nodules like this, but they can be found either on the plant or endophytic, and they can fix nitrogen, and they're not vascular. I just read that somewhere. Let me see if I can put my hands on that one. All right, so this leg hemoglobin is really, this evolved again as a partnership that both partners are involved in building. That's so cool. They're both invested in building this protein complex so that they can solve this problem of we can't have too much oxygen around the nitrogenase. And so that is one, it's essentially sequestering the oxygen, if you want to think of it that way, giving enough oxygen availability for respiration, but not too much.
Sorry, I know so many questions today, but the bacteria, like whatever species this is, they are aerobic, right? So they need oxygen
Yes.
as well.
Yes.
So in the soil, if the plant is making the globin, like how does the bacteria control, like not messing with this enzyme when it's like free living?
So sometimes when they're free living, they won't do it. They won't do nitrogen fixation.
Okay.
They just won't, because it might just be a problem where it's going to end up, their nitrogenase is going to be toast, So some bacteria that do symbiotic nitrogen fixation, if they're by themselves and not symbiotic, they just won't do it. And then there are some free living ones that, this next one. There are some free living bacteria that aren't symbionts. They just naturally, I'm always free living. So cyanobacteria are a good example of that. They just hang out and they're not in the rhizobia group where they form this symbiotic relationship with the plant, but they do nitrogen fixation. And so, cyanobacteria, they produce oxygen when they're doing photosynthesis. So it's like, what in the world? How can we have cyanobacteria fixing nitrogen if they're actually making so much oxygen? How do they keep the nitrogenase enzyme safe from too much oxygen? So they evolved heterocysts, which are special cells that I'll show you a picture. I don't have it yet. I have the next one. So cyanobacteria grow, they have these so-called, the normal cells we call vegetative cells, and they grow like pearls on a pearl necklace, just imagine that. And then one of the pearls is, instead of being a vegetative cell, it's a heterocyst. And then you crack that open and look inside and go, what's going on in this special cell? It's fixing nitrogen in there. But all the other cells are just going about, I'm photosynthesizing, I'm producing a bunch of oxygen. But that one special heterocyst says, well, I'm not going to do any of that because oxygen will kill, denature the nitrogenase. So let's just devote this special compartment to fixing nitrogen. And then we can, by diffusion, get the fixed nitrogen to the other cells. And so they compartmentalize and structurally isolate where the nitrogenase is away from all the other oxygen. Really cool adaptation, just so fascinating. And so even though they're pumping out oxygen during photosynthesis, they can still have their dinitrogenase enzyme up and running. I just wanted to show this. Anyone who is familiar with animal or human phys, you've probably seen a hemoglobin or myoglobin. This is the three-dimensional structure of the protein complex with the alpha sheets and beta coils and blah, blah, or alpha beta sheets and alpha helices and beta sheets, I think is the word. Yeah. So here's the subunit complex here of, with the idea of hemoglobin, and then leg hemoglobin. Look at that. It's so similar, so similar. And with, if you're wondering what a heme is, that's what the heme looks like, the heme structure with the iron chelated in there. So leg hemoglobin, that's what they're using to bind oxygen and then just slowly release it as needed so the bacteria can have it for respiration, the plant cells can have it for respiration. So has anyone in here eaten a Impossible Whopper? So you've eaten leg hemoglobin. That's what they put in that patty. So the Impossible Whopper, for those of you who may not know, it's totally plant-based patty that looks and tastes and smells like beef, like hamburger. And actually, I was at a plant biology conference in California 2 summers ago, and the company, the Impossible, whatever the company is, I can't remember the name of the company, but they had a big, huge symposium on how they went about scientifically creating this. It was fascinating. And I was so tickled when they said one of the big things, they did a lot of case studies with humans, like what do you like to smell when you eat a beef patty? Yeah, blood, right?
Yeah.
Blood is part of it. The taste of blood, as even cooked blood, your taste buds can pick up on that, and that signals to your brain, I'm eating meat. And so they're like, how can we do that so that it's faithful to that, so that people think they're really eating meat? Leg hemoglobin. They did it. I'm like, that is just genius that they picked leg hemoglobin, because it's pink. When you cook it, it'll look like blood that's been cooked, and it has the iron in it, so it tastes like, get that mild taste. Just a fascinating symposium. They went on and on and on with all the other stuff that's involved in making the patty. Just a really, really, it was so fun to listen to. And I just love their experimental process. They really got into it, getting all into the sensory, every sensory aspect when you eat food. They had to consider that very, very carefully in order to create something that could so closely mimic. So if you ever want to take a break from studying and Google that, there's some nice articles about how they went about doing it. It's really, I really like their scientific method. I thought they put that to good use, and they're making a lot of money. Here's the heterocyst idea. So this is a cyanobacteria, and the vegetative cells are busy doing photosynthesis, pumping out a bunch of oxygen. And the heterocyst says, I'm not going to do that. I'll just fix nitrogen right here, and I'll let it diffuse out to the rest of the cells. Okay. And then I updated the slide set because I wanted to update this last slide, and I'm not going to go through it right now, but if you want to read it, because I was taught that having too much nitrate, which plants love as a nitrogen source, is bad for you, like bad for our health. And I grew up with that. Now I just read in a 2018 paper where they're saying, wait a minute, maybe not. So these two are, the first bullet point is what we used to think. The second bullet point is raising some questions about that and saying it might not be as bad as you think, and in fact can actually be good. So I was stunned. I read that this summer. I'm like, oh, geez. Here I've been told forever nitrate's bad for us and it can be carcinogenic in too high of quantities. And actually many countries will limit, they'll test a tissue sample from plants that you're about to eat. And if it scores too high in nitrate, they say, nope, you can't sell it. So I just wanted to make you aware that this is an issue, a long-standing issue. They think it's dangerous. Now they're saying maybe not. So I just wanted to put that out there in case you're curious about what you're eating and nitrate levels. Okay.
So does that differ from the nitrate levels that are high in processed meats, or is that like a similar finding?
So yeah, so what I was told is like nitrate in high quantities is found in processed meats. It's so because nitrate actually is a way to inhibit bacterial growth. If you spike the meat with a lot of nitrates, what it's actually doing is killing the microbes. So if it's killing the microbes, it's like, well, what does that do to us? And so there was this concern that too much nitrates in our body can lead to carcinogenic conditions. And so then they said, let's stop doing, let's be aware of how much nitrate. So for example, when I was pregnant, I didn't eat any processed meat because they're worried the nitrates would get to the fetus and cause issues. So so having said that, too much of anything can be bad for you, right? So obviously there is a threshold, but I was excited to see that at least in this study and several of the studies they cite in this paper, they're saying, wait a minute, let's be a little bit more cautious about, nitrate's horrible for you, maybe not. High doses, yeah. So I still don't eat processed meat to, frequent levels. I don't do that. And I try to avoid high nitrate levels, but now I'm not so worried about nitrate in general after reading that. So anyways, I just thought I'd put that out there. for anyone.
You
who-
sent out a link to this paper?
It's cited in the notes. If you go back into EduCat, I just posted an updated slide set on biological nitrogen fixation. It's the very last slide in that slide set, and in the notes section, it'll give you the reference. It's there.
Okay, thanks.
Yeah. All right, we ran out of time. We're done with biological nitrogen fixation. Very fascinating, but now we're going to get into light and chlorophyll and the light reactions next. Why won't you?
So for the cyanobacteria, are they forming colonies, or are they like not unicellular, or are they like separate chambers where they have the heterocyst and not like an actual cell? You know what I mean?
The heterocyst?
So
What-
you're talking about the cyanobacteria.
Yeah.
The heterocyst. But you said they're like different cells. Is that like a colony of
Yes.
bacteria?
A colony, several cyanobacterial cells stick together into a colony.
Oh,
Yeah.
Yeah, because I've only ever seen the pictures of them in strings. So
Yep.
I guess I assumed that like that was all one.
So several
organism.
cells, yeah, all one
Yeah.
organism, several cells of it. So they're,
Are all bacteria not unicellular then? Or
they're,
is
well,
this
each cell is unicellular, it's just that they stay connected.
So are they like genetically different, like each of the blobs?
Well, I think like when one cyanobacteria goes through cell division, they just stick to each other. Then that one goes through cell division, they stick to each other and they form, you know what I mean?
Yeah.
So they're like progeny cells stuck to each other.
Okay, I'm gonna look it up, but sorry, I'm doing it.
Yeah, I would definitely